Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. So, in our novel drug delivery systems, we are discussing transdermal drug delivery systems. In our previous lectures, we started with the introduction of transdermal drug delivery systems, and also we have seen in our previous session the factors affecting the transdermal permeation. We have seen the physicochemical properties of the drug molecules, and also we have seen the physicochemical properties of the drug delivery systems, and then we have seen the biological factors, that is skin will influence the permeation of the drug molecules which are applied through transdermal patches. So, today we are going to discuss on the interesting topic, that is the components of transdermal drug delivery systems. So, it is very important because these components will help in the permeation as well as controlling the release, and it will help in the long time storage of the drug. So, the components or selection of the components while designing of transdermal patches is very important. So, here we have the various components, that is of course, the first is the drug, then polymer matrix, skin permeation enhancers, pressure sensitive adhesives, and also rate controlling membranes, and the backing laminate or membrane. Then we will see there are the other components. Now, you can see in the pictorial representation. So, this is the skin, it has a various layers, and so the topmost layer is the backing layer, and then it represents with the blue color drug, and also here we use some of the membranes which will control the release, and then this is the adhesive and the liner. So, in this way we need to apply the transdermal patches on the skin, but this liner will remove before administration, then adhesive membrane will stick to the skin, and then the drug which we release through the rate controlling membrane which will control the release, adhesive membrane will adhere or which will retain with the skin. The backing layer will help to avoid all type of the entry of the moisture and the dryness of the molecule or dryness of the patch. So, it will help for the protection purpose. So, we will see one by one. So, already we have discussed about the drug properties, still we will consider few points here. The first point is the dose that should be below 20 mg, that is a major limitation of the transdermal patches, and here these drugs need to be potent enough within this range. You cannot take the drugs which are 500 mg or 200 mg and even the 100 mg also, right. So, yes, you can use some of the strategies, you can improve the drug loading, but still we need to select potent drugs which has a low dose. And yes, we have seen the lipophilic factors and the partition coefficient or the fat solubility, the log P should be within 1 to 3 or that should be more than 1, which is eligible for the preparation or designing of the transdermal patches. And the molecular weight, that should be less than 400 Daltons. We can remember Lipinski rule of 5. that is important for the absorption of various drugs for the oral route. So, here also that should be less than 400 Daltons, which are efficient for the transdermal patches. Melting point should be low for better diffusion, because if you have the very low melting point, at that the temperature at the body or on the skin, that will also help for the diffusion. Skin irritation, drug should not have any type of irritation to the skin. Immunogenic, yes, your drug should be non-immunogenic, that should not trigger any type of immune response after the administration. And second component is polymer matrix. As we have studied thoroughly on this topic, the polymers, also we have discussed in the designing of control release drug delivery systems. We have discussed what are the matrix systems, what are the reservoirs, and yes, here also we can use polymers. They have major role in the control drug release. The selection of polymer will ultimately influence the release of your drug from the device or from the patch to the skin. So, the life of the drug will ultimately that depends on the selection of the polymer and usage of the polymer. So, that should be chemically stable, and your polymer should be non-reactive and easy to manufacture, and that should be cost effective, and that should not degrade your active ingredient, that should not influence the release and the performance of your active ingredient, that should allow the drug to diffuse at controlled and the desirable rate. If it not works, then it will not help and that will not fulfill the requirement. Your polymer must not degrade during the storage or use. So, that these parameters are need to be considered. Again, we are seeing the polymers which are used in the transdermal patches. So, types of polymers used, that is first one is natural, which comes from the natural source, gelatin, cellulose derivatives and the chitosan, and the synthetic elastomers, that is silicone rubber, polybutadiene, neoprene and the butyl rubbers. We use these polymers in various applications while preparing of the liners, also in the rate controlling membranes, also we use in the when you want to make in a matrix or reservoir systems with the polymers, and also some type of the backening layers. So, these are the polymers we use in the as a components in the development of the various transdermal patches. And also synthetic polymers, polyvinyl alcohol, PVP, polyvinyl pyrrolidine, PVC, polyvinyl alcohol, also polyethylene. These are the few examples of the polymers which we can use it in the transdermal patches. Now, the third component is skin penetration enhancers. We have already discussed about the skin penetration enhancers in our previous lecture on the factors affecting the permeation. So, we studied these are the compounds which will improve the drug absorption through the skin by reducing the skin barrier resistance. So, we have seen the various mechanisms that will disturb the lipid layer on the stratum cornea, and also it will modify the proteins which are present in the stratum cornea, and also it will help for the solubilization of the drug in the lipidic membrane. These are the various mechanisms will help as in a skin penetration enhancers. Not only that, it will also the help or the disturb the tight junctions in the skin. So, here these are the classifications. The first classification of permeation enhancers is solvents which help to dissolve the drugs and also disturb the skin barrier. That is the ultimate goal of a skin penetration enhancers or permeation enhancers. The best example is ethanol that we use in various even nano formulations as a ethosomes. We have seen, you can refer our lectures in the nano carriers, there we discussed about the ethosomes. We have studied about the effect of ethosomes or the mechanism of ethosomes during the delivery of the nano formulations. Even other solvent like acetone, polyethylene glycol, glycerol, propylene glycol and the dimethyl sulfoxide also works as a permeation enhancers. We use it in the treatment of the various skin diseases. Now, the second class is surface active agents. So, that will disturb the lipid structure of the corneum. So, some of the mechanism is contributing through the permeation enhancers. So, as we know that the surfactant is classified into various categories. Anionic surfactant, these are the examples, the best example is sodium lauryl sulphate SLS, and the non-ionic, the poloxamer or pluronic F68, F127 that comes in the non-ionic, and also we can recommend twin 80 and twin 20 formulations. And cationic surfactants, that is CTAP, cetyl trimethyl ammonium bromide, which contains the ammonium that will give the positive charge, that is the cationic comes from this surfactant. And bile salts, we use sodium taurocholate and sodium deoxycholate. So, these are the some of the agents which will works as a skin penetration enhancers. Now, here you can see the binary systems. Here we use two components. So, when you use single or one component as a propane glycolic effect, the effect is very less. When you use combinations, it will show the synergistic effect. So, that is why it is considered as a binary systems, use of two compounds to enhance the permeation synergistically. So, nothing but 1 plus 1 equal to 3, not 2, because it works on the synergistic mechanism. So, there are the drugs also works as a synergistic mechanism when use combinations, we use generally in the chemotherapy, right. So, similarly miscellaneous enhancers. So, these are the some lecithin, L-amino acids, acid phosphate, phospholipase and the chalonases will also works as a permeation enhancers. These are the biological and chemical agents. We can also add into the formulations which will work as a skin penetration enhancers. The fourth one is pressure sensitive adhesive layer. Now, you can see this material is have the property of the adhesive nature. So, this will contact with the skin. So, this is the one of the crucial or important component in the transdermal patches. So, here you can see the adhesive layer which contain the adhesive materials that will stick with the skin. So, the contact time is ultimately depends on the selection of right pressure sensitive adhesive material. So, what is the function of PSA? Ensure the adhesion of the patch to the skin, and it will act as a matrix for the drug and the excipient. So, that will there are the cases we prepare a adhesive based or drug loaded in the adhesive. So, such case it works as a matrix system, and also it influences the drug release, skin permeation, stability of the patches. So, it has a three different roles: adhesion as well as matrix system also help for the drug release, permeation and the stability of the patch. Now, here we will see some of the desirable properties of any type of pressure sensitive adhesive materials. Your pressure sensitive adhesive material should not irritate or sensitize the skin, as we discussed in other cases also, in case of permeation enhancers as well as drugs also we have seen. So, that should not irritate the skin. Adhere with a gentle finger pressure. After applying with a small force, that need to be adhere on the skin. So, that is the based on that properties we need to select the right component. Should be easily removable without leaving any residues on the surface of the skin, and no unwashable residues will present after the removal of the transdermal patches. There are the materials, so they can leave the residues. Sometimes there are the polymers, or there are the solvents, or there are the adhesive membranes, they may stick and adhere to the skin because of their adhesive properties. So, that should be easily washable and chemically compatible with the drug, excipient and other enhancer. This is an important parameter. So, to select right PSA material, we need to see the compatibility study with the drugs and the excipients and enhancer. Based on that, if your material, if your PSA material is non-compatible, then we should not select. If it is a compatible and there is no problem in the chemical properties of the drugs as well as excipient, so then we can select the right PSA material. Should not interfere with the drug or enhancer permeation. So, that also we need to perform with the various ratios when you take the adhesive membrane, adhesive layer as well as with the drug, and we can make a physical mixture and we can see the release as well as compatibility studies. So, with this we can select a right pressure sensitive adhesive layer. So, these are the some of the examples of the PSA. So, here you can see the type, and here are these are the characteristic properties. So, if you are looking for the hydrophilic properties, then you need to select polyacrylate or acrylic type of the materials, and if you are looking for a saturated or excellent oxygen resistance, then you should prefer polyisobutylene, and if you are looking for stable and non-reactive, then we need to select the silicon based materials, and if you are looking for hydrophobic, strong cohesive strength with low viscosity, then you should select styrene based or rubber based type of the polymers as a pressure sensitive adhesive layers. Now, the fifth point is the rate controlling membrane. Now, in the picture you can see the blue color which is represents the rate controlling membrane. So, this made up of with the polymers that the name itself suggests that it will control the release from the patch. So, it works as a drug reservoir. The drug reservoir is positioned between the impermeable backening layer and the rate controlling membrane. Now, you can see, so this is the drug reservoir. Now, you can see here, and also you can see the impermeable backening layer and then rate controlling. So, in between these two, one that is the impermeable backening layer and the second one is rate controlling membrane. In between, the drug need to be converted into the reservoir system, and that need to be placed in between these two components. So, that will help for the release of the molecules from the patches. So, rate controlling membrane will help for the release of the drug. So, here it will help as a rate controlling membrane. So, these are the various types of the membranes. So, here we use various types of the polymers, that is natural polymers, synthetic and composite membranes, and here is the chitosan is the best example, and also there are the PHEMA materials that can also be used, and ethylene vinyl acetate copolymers, microporous polypropylene and microporous polyethylene can be also used as a rate controlling membranes. Now, the sixth one is this is a backening layer, the topmost one, impermeable backening layer, which will provide the structural support to the patch. So, that will help to provide the support or structural support to the membrane, and this will prevent the drug permeation enhancers. So, the backening layer will prevent the drug and permeation enhancers from leaving the dosage form through the top. So, it will avoid all type of entry into the patch, as well as it will also stop the leakage or movement of the drugs as well as permeation enhancers from the dosage form. These are the key properties, that is impermeable to drugs as well as permeation enhancers, low moisture vapor transmission rate. So, that will avoid all type of the moisture entry, also leavage from the formulation. So, with this you can avoid the dryness of the formulation. Optimal elasticity is important, flexibility, tensile strength also important for your backening membrane. Chemically compared with the drug, enhancers and adhesive and other excipients, the cost effective, suitable for printing and adhesive lamination. So, people, the manufacturers or the commercial products, so people will refer always to give their brand names on the patches. So, that should be suitable for the printing and as well as adhesive lamination. So, here you can use these components, pigmented layers, aluminum vapor coated layers and the plastic films, heat seal layer, this all will help in the backening membrane composition. And in the other components, now you can see here is the backening layer and here is the drug reservoir, rate controlling membrane and as well as adhesive, and the last one is the protective peel strip. And not only that, it contains various type of other ingredients which are plasticizers, solvents, as we discussed here in the first component, which will remove before the application, that is a protective peel strip. So, here to maintain the plasticity and the flexibility, we need to add the propylene glycol, polyethylene glycol, triethyl citrate and dibutyl phthalate. Also solvents we use to prepare the reservoirs. If your drug is insoluble, we can use various types of solvent, and also we can use DMSO also. So, that will help for the solubility, also conversion of the polymers drugs into the reservoir systems. And the protective liners that should cover the patch during the storage and removed before the patch application to the skin. So, before application we need to remove that the protective liner, then we will adhere to the skin. So, that can be made by the polyethylene, PVC, polyester and the styrene based films. So far we have discussed on the various components of the transdermal patches. So, today we have discussed on the components, selection of the components in the transdermal drug delivery systems, and we learned the physicochemical properties of the drugs which are important, and also while selecting the polymer matrix and the skin penetration enhancers, pressure sensitive adhesive membranes, also rate controlling membranes, backening membrane and the other components also very important while designing the transdermal drug delivery systems. So, while designing we need to consider all these parameters based on your drug properties as well as formulation properties. You need to make a right transdermal patches that will help for the treatment of various diseases. So, with this I am going to stop today's lecture. We will see you in next lecture on the various formulation strategies of the transdermal drug delivery, and further we will see some of the evaluation parameters of the transdermal formulations. Thank you so much.